Hello everyone, this is Dr. Marwa Omar. Welcome to this video about the amazing life story of eosinophils. Each one of us has their own sharing life. Eosinophils are no exception. They have incredible stories of their own. Actually, eosinophils are one of the grand team. They are innate cells which act with minutes from the infection. They are feverishly fast and they have no memory. Under normal conditions, usually the eosinophils only constitute a small portion of the circulating leukocytes. Usually it does not exceed 7% of the total leukocytic count. Relationship to different parasites is an important stage in the life story of eosinophils. There is always a special bond between eosinophils and different parasitic infections. Why is that? First of all, eosinophils are large in size. They are much larger than neutrophils, for example. Number two, they have specific receptors for different parasites. And number three, they have phagocytic ability. So that's why, besides allergy, invasive helminthic infections are one of the major causes of eosinophilia, and of course elevation of the eosinophil count in the peripheral blood. Back in 2004, and specifically in neutrophils, the Spider-Man behavior of the glands was first discovered. Such threats which are released from Spider-Man are similarly released from neutrophils and other inflammatory cells upon activation. They are actually composed of DNA fibers which are trying to trap different pathogens. It's considered an innate first-line defense against pathogens which cannot be phagocytosed. Simply, we can name these traps according to the type of the cell. They are called nets or neutrophil extracellular traps in case of neutrophils, and they are called EATS or eosinophil extracellular traps in case of eosinophils. Okay, so what is similar between both of them? They are both DNA fibers composed of cytotoxic histones and granular proteins. They are both fibers which are trying, of course, to trap pathogens that cannot be phagocytosed. What is the process that leads to the release of these threats? Actually, it takes the death of the cell to release such traps. The death of neutrophils, which is called in this case netosis, and death of eosinophils and other inflammatory cells, which is called in this case etosis. So the question now is it a passive programmed cell death, as we call it apoptosis? No, it's an active cytolytic cell death. It does not involve fragmentation of the DNA, as we can see in apoptosis. It involves disintegration of the plasma membrane and the nuclear envelope. So that is the main difference between such an active process and the usual apoptosis or necrosis. The main difference between netosis and etosis is something related to the granules. In case of netosis or death of the neutrophils, the granules undergo intracellular disintegration, and so they become sticky and adhere to these traps, become part of these traps. This is not the case in eosinophils. In case of etosis or the active cytolytic death of the eosinophil, most of the granules remain intact during the process, and so this results in the release of free extracellular granules. Recently, in 2021, a review article was published by a group of Japanese and American scientists who have created this amazing cartoonic depiction to summarize the life story of eosinophils, and they called it EOSMAN. Okay, the story begins inside the bone marrow, as you all know. The cells originate from the pluripotent hematopoietic stem cells. They are terminally differentiated, which means that they are short-lived. They circulate inside the blood before they migrate to the tissues, especially of the GIT, so as to keep homeostasis. The trafficking and migration of eosinophil from the circulation to the peripheral tissues is a multi-step process which involves rolling, tethering, firm adhesion to the vascular endothelium, transmigration across the vascular endothelium, of course, of the lung, GIT, and skin. In cases of allergy and invasive helminthic infections, the eosinophils are recruited to the site of infection or inflammation. First of all, they have to warm up and repair itself, which we call it priming. Then it begins to release its toxic weapons, whether it was cytokine release or degranulation and the release of its granules. We have mentioned before the process of etosis, which is considered an active cytolytic death of the eosinophils and other inflammatory cells. We have mentioned that it's different from the passive apoptosis, that it's active process, it does not involve fragmentation of the DNA, and it involves also, in case of eosinophils, the release of the extracellular granules and the intracellular circuit-lighting crystals. We have mentioned that this process is exhibited by the eosinophil to the degree that it makes it sacrifice itself in order to trap pathogens that cannot be phagocytosed, and we have mentioned that it's an innate first defense line. So, we have mentioned that eosinophils are actually short-lived. The prolonged survival of the eosinophils actually is related to certain cytokines which is released from the eosinophil itself, as well as especially from the T helper 2 cells. Such prolonged survival contribute to the severity of allergy, to the severity of inflammation as well. From the clinical point of view, the aggregation of the extracellular traps, or as we have called it, EATS, actually it was related to the severity of allergic conditions, whether it was asthmatic attacks or allergic rhinosinusitis. So, if we could target these aggregates of eosinophils, I think this would be a good therapeutic potential for such clinical conditions, whether it was allergy, asthmatic attacks, and other clinical conditions that incriminate eosinophils in histopathology. And so we got to the end of the story of such an amazing cell. Thank you so much for listening.